Hi everyone, welcome to the second unit the second lesson in our biology unit, the development of the cell theory. In this lesson, you will learn about spontaneous generation and the cell theory. I do want to take this moment to point out, in these videos, you see, I am kind of touching on the main points, but it's important for you to know that I don't cover every bit of knowledge that's in the textbook. So it's important to watch this video, but to also read the textbook section that is related to this video. And if you're on the workspace, I always say watch the video and read the text, and I've linked the text right there for you. So you really have to make sure you're not just relying on, like, the videos tell you the main part, but there's a lot of information that you also need to know that's in the textbook. So make sure, watch the lesson video, read the associated textbook. I just want to say that before I get started. Moving on to spontaneous generation, you see, there was this theory where basically people thought that life could just magically appear in various situations. It would just spontaneously emerge. Like, how could it just happen? So scientists, try to figure out, well, how can it be that you have an object and all of a sudden we find living material there? What's going on? Maybe something's dead and all of a sudden we come back and there's flies around. Like, how could it be that we have something non-living and then we come back and now there are living things present? So, spontaneous generation, the idea that life could emerge spontaneously from non-living matter, was widely accepted from the time of the Romans through the 19th century. So what they thought, for example, if I use this picture right here, you have a piece of meat, it is non-living. It was widely accepted that life could emerge from non-living things. No big deal. In your textbook, it talks about a mice example. I'll read it to you here. It said here, it was generally accepted that to produce mice, one simply had to put sweaty underwear and husks of wheat in an open jar. About 21 days later, the sweat and husks would combine and change the husks into mice. There you go. We literally have life kind of combining or appearing, spontaneously generating out of non-living material, and that was accepted. In 1668, Reddy questioned the notion of spontaneous generation. Now here's what he did. If you look at this lab right here, so we have pieces of meat in all of them, and let's look at the differences here. It's key, the differences. On the left, right here, we have an open jar. So it looks like the meat is openly accessible to the outer environment. Air, anything else can get to it. If you look right here, in this case, we put the meat into sealed jars where nothing can get in or out. And on the third example, we have jars that are covered with cloth netting. So let's look at the results here now. In the open jars on the left, so you put the dead meat in, and if you come back, we found that the dead meat was covered in flies or what have you. If you put the dead meat into jars that are totally sealed in the middle, when you come back later, there are no flies surrounding the meat. They're not inside. And on the third example, we put the dead meat on the inside and cover it up with the cloth netting. Again, we find that the flies are not in there. Now, it's obvious that the air can get in or out here, but the flies are not in there. So the maggots, the flies, were only found in flasks that were open and accessible to flies to lay their eggs. So you see what this is proving here now, everyone, is that we have this dead material, and the maggots, the flies, they weren't just appearing out of nowhere. How did anything grow on the meat? Well, the new life only grew on the dead meat when the flies were able to enter in and lay their eggs on the meat. And then, of course, you get the maggots. So you see, this is disproving spontaneous generation. You can't just have life emerge from non-living things. The only reason the maggots grew here is because the flies were able to fly into the jar, lay their eggs, and then, of course, the eggs would hatch and grow and so on and so forth. In these two cases over here, maybe the flies tried to get in there, but they couldn't. There was no laying of the eggs. So we had no new generation of life. So spontaneous generation was disproved here. The second example is Louis Pasteur. Now, this is important, and this is also going to be on your quiz and exam. And he kind of, for once and for all, this was the finality of it. The spontaneous generation debate was put to rest. Now, the textbook describes it here on the left in these words, but I'm going to show you, I think this example over here is much better. So what they did is they took this broth, okay, and it's full of nutrients, and they heated it. And what they did is by heating it, just like at home when you boil something, you're trying to kill all the microbes, okay? So what we know is we take this nutrient-rich broth and we heat it. We superheat it, and we know that we have killed all the microbes. So there's nothing living in there. Now it's important to notice that you have this curved neck, like that. And that really served an important purpose. What that means is what the textbook goes on to talk about is that this curved neck stopped any microbes or living things from actually reaching into the nutrient-rich broth. So for example, if you take the fly might want to lay its eggs, but you see the fly would fly in and get caught right there. Nothing living could get all the way down to the nutrient-rich broth. So that means, essentially, this nutrient-rich broth was left untouched. All the microorganisms, the microbes in there had been killed. So there is nothing living in here, and nothing living externally could come in and lay eggs or reproduce, etc., etc. And what they found is for an entire year, nothing grew in here. It's full of nutrients. It's full of food. It's not heated anymore, but nothing was growing. So here we have an example of where this non-living material in here, nothing's just spontaneously growing out of it. So what they did after the 1 year of time is they break off or remove this curved neck. Now it's a straight shot, easy entry into this nutrient-rich broth. And what they found is once this was left exposed and things could kind of get in here, it happened right away. The nutrient-rich broth all of a sudden has tons of microorganisms growing in it. So this is the final example of showing that spontaneous generation was false. You cannot have just life just burst from something that's non-living. The only way that you can see new life from non-living things is when an external source of life can reach that dead material, lay eggs, so on and so forth. You need to be familiar with this experiment for your quiz and exam. The cell theory, just some kind of leading up here to the cell theory. The invention of the microscope allowed scientists to magnify objects and examine the microscopic world. As a result, cells were discovered. And I talked about that in the last video. Think about it. Before the microscope, scientists weren't able to look and see this small world and truly understand it. What is a cell? The smallest functional unit of life found in all living organisms. In 1833, Robert Brown identified an important cell structure, the nucleus. And I believe this is what he saw right here. Okay. Brown found an opaque granular spot within the cell. So again, I could zoom out, but this is the cell, and he found these opaque granular spots. In 1838, Scheidlin observed that all plants were composed of cells, and he proposed that the nucleus was in fact the structure responsible for the development of the remainder of the cell. So that's really interesting here. I want you to remember that. Scheidlin observed plants. He was looking at plants. And he was looking at cells, and he found there was a nucleus in the cell. He also determined, or believed, that the nucleus was responsible for essentially the operation of the entire cell. Now this person, Scheidlin, discussed his work with Schwann. Now he was studying animal cells. Schwann believed that there were similarities between the plant and animal tissue. So when Schwann searched for the opaque spots in animal tissue, he found structures that resembled the nucleus that had been found on plants. So essentially what's happening here is, so Schwann comes along, he looks now at animal cells, and he goes, holy cow, I can see those same type spots right there. So here's what they figured out. They go, holy cow. Schleidlin and Schwann proposed that all plants and animals were composed of cells, and the cell was the basic unit of life for all organisms. They had a cell, they had a nucleus. So this is the first time where people really realize plants and animals both have these cells and nucleus, and how important they are to the function of all living things, plants and animals. This leads now finally to the cell theory, and yes, there is a question coming up. It's at your quiz, your final exam, where they say which one of these is not. So am I telling you have to know these? The answer is yes. The cell theory states all living things are made up of one or more cells. And okay, so all living things are made up of one or more cells. Number two, all life functions take place in cells, making them the smallest unit of life. So within it, so if you have a cell, if we could zoom in, there is so much going on inside of a cell, and we'll look at that coming up. All cells are produced from pre-existing cells through the process of cell division. Again, you don't just have a cell and then create a new one magically. All cells come from previous cells. You must know the cell theory and these 3 points.